Exploring apoptosis.
Nasopharyngeal carcinoma, or NPC, has been a widely studied issue among the world, but there are significant geographical disparities in the prevalence of this cancer. Specifically, the burden of NPC is most prevalent across Asia, accounting for 84.6% of the world's incident cases of NPC according to the Globocan 2018 estimates. Currently, chemotherapy is the primary option for treatment against nasopharyngeal carcinoma, in specific to advanced or metastatic stages. Application of cisplatin-based methods are commonly used to hinder the growth of cancer cells. However, its therapeutic outcome may be impeded due to its toxicity and resistance. Therefore, investigation into new effective treatments formulated from natural compounds may have promising effects against cisplatin-resistant NPC cells. Asiatic acid, or AA, has been discovered to have various pharmacological properties such as anti-inflammatory and antioxidant. Moreover, it is involved in various molecular mechanisms in relation to its neuron cardioprotective, antimicrobial, and anti-tumor effects. Many studies have reported the anti-cancer properties of AA. One research proposed by Hughes et al. Revealed the anti-cancer effects of AA in breast cancer through cell cycle arrest and mitochondrial apoptotic pathways. Yet, the effect of AA remains unclear for treating NPC. Thus, examining into the cytotoxic effects and mediated mechanisms of AA can assist in reducing the cisplatin-resistant NPC cell lines such as NPC039 and NPC-BM. This can be examined by first performing the Western blotting technique.
This technique is employed to detect specific proteins within a complex mixture extracted from cells. In medical science, it is used to assess protein expression, analyze protein modifications, and verify the presence of particular proteins in a sample. It also plays a role in clinical diagnostics, aiding in the diagnosis of diseases such as HIV/AIDS, hepatitis B, and Lyme disease. In this experiment, the NPC039 and NPC-BM cell lines are treated with varying concentrations of acetic acid at 0, 25, 50, and 75 micromoles. Then, the cells are washed with cold PBS to remove extracellular contaminants and maintain the cells in an inactive state, and lysed with Ripper buffer to release cellular proteins. BCA protein assay and a standard curve was used to determine the total concentration of proteins present in the supernatant of the samples. Proteins in the sample are separated via gel electrophoresis to analyze MAPK pathway proteins and compare their expression and phosphorylation between normal and cancer cells. Then, they are transferred from the gel to a PVDF membrane via an electric current going from a negative to a positive electrode. The membrane is then blocked with 5% non-fat milk in TBST to prevent nonspecific binding of antibodies to the membrane. The membrane is then incubated with primary antibodies that specifically bind to their target proteins. These are the primary antibodies and their corresponding target protein. Then, the membrane is rinsed and incubated with HRP-linked secondary antibodies. This is important as the secondary antibody will recognize and bind to the primary antibodies, amplifying the signal and allowing for the detection of the target proteins. Lastly, a chemiluminescent HRP substrate was used to visualize the immunoblots. Chemiluminescent refers to light emitted because of a chemical reaction, such as a HRP-linked secondary antibodies reacting with a HRP substrate. This is an example of the immunoblots visualized. Western blotting has its limitations, including dependency on primary antibodies, which are expensive, ranging from $100 to $2,000. Without these antibodies, Western blotting for the desired protein is impossible. Technical errors in primary antibody use, like incorrect concentrations, can affect result reliability. Also, many antibodies exhibit off-target effects interacting with unintended proteins, complicating specific protein analysis. During protein transfer to the PVDF membrane, proteins smaller than 10 kilodaltons may not be retained, and those larger than 140 kilodaltons may not transfer efficiently. These issues can affect Western blot accuracy and compromise result validity.
In this article, there were two main key findings associated with the Western blot analysis technique used. Key finding 1 explores the caspase activation pathway, while key finding 2 explains the cellular mechanisms used in AA-induced apoptosis. The study uses NPC039 and NPC-BM cell lines. For the NPC039 cell lines, they are derived from primary nasopharyngeal carcinoma tumors, which represent the characteristics of NPC cells in their primary state, the nasopharynx. For NPC-BM cell lines, they are derived from metastatic nasopharyngeal carcinoma cells that have spread to other regions or organs of the body. Key finding 1. Caspase and apoptotic protein pathway. Caspases. The researchers investigated cleaved forms of caspase 3, 8, and 9 in AA-treated NPC cell lines. In cis NPC 039 cell lines treated with AA at 50 micrometers, there was a significant increase in the expression of caspase 3, 8, and 9. In cis NPC BM cell lines, AA had to be at a higher concentration of 75 micrometers, which then increased expressions of caspase 3, 8, and 9, suggesting metastatic cells were more resistant to capsase activation. Caspases are enzymes that play a crucial role in orchestrating apoptosis through two main pathways, the intrinsic or mitochondrial pathway and the extrinsic or death receptor pathway. For the intrinsic pathway, mitochondrial damage leads to the release of cytochrome C. Cytochrome C then binds to APAF1, forming the apoptosome and activating caspase 9. Caspase 9 then activates execution of capsases, such as capsase 3 and capsase 7. The extrinsic pathway uses death receptors that engage with ligands, such as FAS-L or TRAIL. The death-inducing signaling complex forms, activating capsase 8. Capsase 8 then activates execution of capsases and amplifies the apoptotic signal via the mitochondrial pathway. Pro-apoptotic proteins. The researchers also investigated pro-apoptotic proteins BAX and BAK. In cis NPC BM cell lines, BAX and BAK expressions increased by 183% and 137% respectively at 50 micrometers, whereas BAK expression decreased. In cis NPC cell lines, BAX and BAK expression increased, with 50 micrometers and 75 micrometers of AA treatment. So what does this mean? BAC and BAX proteins are pro-apoptotic proteins that promote cell death by regulating mitochondrial membrane permeability. The study observed that AA treatment increased the levels of BAX and BAC in certain NPC cell lines, which supports the induction of apoptosis through mitochondrial apoptotic pathways. In summary, AA induces apoptosis in NPC cells by both activating key enzymes involved in cell death and altering the levels of proteins that regulate the mitochondrial pathways of apoptosis. Key finding 2. Mechanisms of AA-mediated apoptosis. Moving on to our second key finding, the researchers explored mechanisms related to AA-mediated apoptosis by analyzing the expression levels of stress response proteins and cell survival proteins through the Western blot analysis. In NPC 039 cell lines, P38 and JNK1/2 proteins showed a significant increase in phosphorylation or activation in a dose-dependent manner when treated with AA. AKT showed a significant decrease in phosphorylation or activation when treated with 50 micrometers and 75 micrometers of AA. As for ERK1/2, there was also a decrease in phosphorylation of ERK1/2 in the group treated with 50 micrometers of AA. In cis NPC BM cell lines, there was an increase in phosphorylation of P38 at 50 micrometers and 75 micrometers. However, GNK1/2 levels were not as profound as levels in 039 cells, again showing the resistance of metastatic cells against stress response protein kinases. There was a significant decrease in expression of ERK1/2 and ARK levels in the 75 micrometer treated group. So what does this mean? Since P38 and JNK1/2 are involved in stress responses and inducing apoptosis, their activation indicates that AA may be promoting apoptosis through these signaling pathways. All the key findings mentioned relate to one another in order to initiate apoptosis successfully in NPC cell lines.
This experiment helps advance modern-day medicine by offering new cancer treatments with less side effects. It is a good indication to continue researching about natural compounds like asiatic acid to be potential candidates for anti-cancer pharmaceuticals, which allows us to expand our current treatment options. For example, some distinct results observed from the cell lines cis-NPC-BM and cis-NPC-039 suggest personalized treatment in which the cellular characteristics of cancer cell lines are biopsied and strategies can be tailored to each patient's cancer. As this is only the first study to demonstrate the AA-induced apoptotic pathway in human cisplatin-resistant nasopharyngeal carcinoma, there is still additional research to be done, as it is shown that some results were unable to remain consistent with previous literature. With the information being limited, necessary explanations on variations in the results are to be sought for.